From my background, I am an analytical chemist, now already working for more than 1 decade in the field of LC-MS-MS-based mycotoxin analysis. But let's now just directly move into the first major topic of this talk, namely the link between climate change and mycotoxin formation. In our last webinar, I have talked about the basics of mycotoxins. So how they are basically formed, which kind of mycotoxins are there, and what are the analytical toolboxes in order to tackle challenges coming along with mycotoxin contamination. Today, we will dive a little bit deeper into this topic and look how rising temperatures and also changing rainfall patterns will influence the fungal contamination in our crops. So climate change is not just an environmental issue. It directly affects crops and also the toxins they may carry. Rising temperatures, shifting rainfall, and also more extremes create conditions that favor fungal growth and also the toxin production. And here on this slide, we can see how global warming relates linearly to cumulative CO2 emissions, illustrated with the so-called shared socioeconomic pathway scenarios, which basically combines the societal development with emission trajectories. So that means, in short, SSP values, lower values like 1 to 1.9, represents very ambitious climate actions, whereby these scenarios with a higher number, 5 to 8.5, represents high emission fossil fueled growth. The key point is the strong linear relationship between the cumulative CO2 emissions and also the global warming. Even moderate emissions, like in a moderate scenario like SSP 2 to 4.5, result in a warming of around 2.7 degrees Celsius by the end of our century, which is already above the 1.5 degree target. And such a level of warming is also enough to significantly alter fungal ecology. And this isn't just a theoretical number, it's essentially what current projections anticipate when factoring in our present efforts to combat global warming. So what does this mean for agriculture, especially for maize, one of the world's most important crops? So maize yields are highly sensitive to global warming. We can see that in this picture. Even a moderate rise of around 2 degrees Celsius cause significant yield declines in many regions of the world, as we can see, especially here in the middle and the last picture. And if the temperatures increase by more than 4 degrees Celsius, global losses could be really severe. Regions like in Africa, but also South America and also parts of Asia are particularly vulnerable, what the projections shows us. The point is, when maize is stressed by heat, drought or other climate variabilities, it also becomes more vulnerable to fungal infections. And this is actually how the risk of mycotoxins enter this picture. And that basically brings us directly to the first toxin we need to talk about, namely aflatoxin B1. So even under moderate warming, aflatoxin B1 is becoming a serious problem in Europe. So traditionally, aflatoxins were only a concern in very dry and hot tropical and subtropical regions, but risk areas are now shifting northward. A key study in this field, conducted by Paola Battigliani and colleagues from Italy, used, for example, a mechanistic aflamase model and linked it to crop flowering and harvest dates, simulating 100 years of daily weather at over 2,000 sites across Europe. And the results show that even a moderate increase in temperature of around plus 2 degrees Celsius, the risk of maize exceeding the European unit legal limit of 5 microgram per kilogram increases significantly. So today we have around 20% of these sites which already surpassed this limit, and global warming will make the problem even worse. So how worse? We can see that on this map, which shows the projected aflatoxin B1 hotspots in Europe. The highest risk is in the Balkan region, but also parts of Italy and Eastern Europe. You can see it clearly here. While Northern Europe is still relatively safe, I would say, but ongoing warming may also create new risk areas in the future, especially with elevated temperatures, which you can see here in the last slide. So generally, we face here a double challenge. Maize yields are expected to decline on the one side under warming scenarios, and on the other side, the remaining maize is increasingly exposed to aflatoxin contamination. This means, in other words, less crop and higher mycotoxin risk. Wheat, on the other side, is largely unaffected by aflatoxins. You can see that in the lower pictures. So maize can be really seen as the main concern for climate-driven aflatoxin risk. And the consequences are clear. A smaller, safe maize supply could cause also economic losses and greater exposure risk, highlighting the need for proactive monitoring and also mitigation strategies across Europe. And these scenarios are not just some predictions, they are already happening. And a clear example is the recent aflatoxin M1 crisis we had to face in Serbia. So what happened in Serbia? The extreme drought and heat in the year 2012 created, so to speak, ideal conditions for the Aspergillus flavus, the fungus which is mainly producing aflatoxins, leading to severe maize contamination. And by early 2013, milk samples showed aflatoxin M1 levels far above the European Union safety limits, reflecting direct transfer from contaminated feed. So the government in this case responded by destroying milk and also temporarily raised the legal limit. And of course, this crisis caused major disruptions in the dairy sector, and Serbia became increasingly dependent on imported milk powder, which was also commented in a Serbian newspaper this year with the title, "The Collapse of the Serbian Dairy Industry When Water Turns into Milk." However, it also raises an important question. How does the contamination in maize actually translate into contamination in milk? And the answer lies in the cow's metabolism. So after ingestion of contaminated maize, aflatoxin B1 is adsorbed in the gastrointestinal tract and transported to the liver. So there it undergoes a phase 1 metabolism, mainly via so-called cytochrome P450 enzymes. The key step is here the hydroxylation at the 9-alpha position, converting the aflatoxin B1 into the M1 version, which is then basically excreted via bile and urine, but most importantly, secreted into milk, where it remains chemically stable. Typically, about 1 to 6% of the ingested aflatoxin B1 is then converted into aflatoxin M1. And while this fraction seems a little bit small, continuous intake of contaminated maize can result in milk concentrations exceeding the regulatory limits. And this makes the maize milk pathway a critical link also for human exposure. So the Serbian aflatoxin crisis was, so to speak, a real wake-up call. Between 2013 and 2015, milk had to be pulled from the shelves. Also, farmers and producers suffered big financial losses, and the consumer trust collapsed. The milk consumption in Belgrade alone dropped by over 25%. The exports went down, and the processors were forced to cut prices by around 15 to 20%. Meanwhile, in countries like New Zealand, the United States, and also Germany, the farm gate prices kept rising with the global market. But in Serbia, the prices stagnated or even fell. The net exports dropped as well, making Serbia some kind of a standout as a negative example. And this actually shows how vulnerable, especially the dairy sector is. So climate extremes and fungal outbreaks don't just threaten food safety, but they can really trigger market disruptions, and also major income losses. And it was one of the first clear signals in Europe, I would say, that climate change is not a far off problem. It's already reshaping both food safety on the one side and also the economic stability on the other side. And when we talk about climate change, we not only talk about heat and drought, it also brings more moisture and heavy rainfall. And this is where actually Fusarium comes in, a fungus that thrives in humid and moderately warm conditions. So from scientific reports, we know that heavy rainfall events increase by about 7 to 8% with every degree of warming. In Austria, for example, such extremes have already risen by around 15% in the past 40 years. Similar trends can be also seen in other countries like in Denmark, China, or also in the United States. And I think the takeaway here in this case is clear. With more frequent and intense rainfall, the risk of Fusarium infections will grow in Central Europe, but also in other regions worldwide. And to see how this unfolds in practice, let's look at the disease cycle of one of the most relevant crop diseases, namely Fusarium head blight. Fusarium head blight starts basically from fungal spores that survive winter on maize residues or in soil. So in spring, these spores infect wheat spikes during the flowering stage. So the infection thrives when it's wet for extended periods, and also the temperatures are moderate between 15 to 30 degrees Celsius. Under these conditions, the fungus spreads quickly, causing spikelet death, yield loss, and the buildup for mycotoxins, especially for deoxynivalenol, zearalenone, fumonisins, but also T2 toxin. So climate change is basically making these conditions more common, and warmer temperatures, more frequent rain, and higher humidity during the wheat flowering create ideal conditions for Fusarium, and increasing both the frequency and also the severity of the outbreaks. And observations actually show that these Fusarium infections, and especially the deoxynivalenol contamination, are now more common across Europe. So in northern countries like the United Kingdom, but also Sweden, Denmark, Scandinavia, infections are frequent, but the toxin levels are usually moderate. On the other side, in southern countries like in France, Italy, Romania, the toxin concentrations are higher and sometimes also exceed the European limits. So these differences actually reflect climate and crop management. So wetter and moderately warm conditions in the south favor severe outbreaks, while the northern regions see frequent but milder contamination. And the main point is that Fusarium risk is no longer rare or just a localized phenomenon. It is persistent, varies by region, and needs active management to protect both yields and also, of course, food safety. So the regional patterns of Fusarium contamination and this northward shift of aflatoxins show that climate change is definitely reshaping mycotoxin risks in Europe. And this actually matters beyond the field. So because mycotoxins link climate variability, human health, and also economic stability. Chronic exposure to deoxynivalenol and aflatoxin B1 is definitely a concern, not only for humans, but also for livestock. And multiple co-occurring Fusarium toxins may even amplify these health risks. From an economical perspective, these outbreaks are, of course, very costly. So in the past decade, tens of millions of tons of wheat were downgraded from food to feed, causing billions in losses. And regionally, southern countries face higher risks due to warmer and wetter conditions, while the northern countries see the lower contamination, but a higher prevalence. So in total, a very challenging situation. And to make things even more complicated, there is an increasing challenge with this co-occurrence of multiple mycotoxins in the same sample. Large surveys show that climate change is creating complex contamination patterns with a mixture of regulated toxins like zearalenone, deoxynivalenol, fumonisins, and the so-called masked mycotoxins, such as deoxynivalenol 3-glucoside, as well as emerging mycotoxins like eniatines and biovaricin. So many samples now contain multiple toxins at once, reflecting how fungi and environmental stress actually interact. And when multiple toxins occur together, their effect can add up or interact synergistically, leading to a greater risk for humans and animals. So this makes traditional single toxin monitoring more or less a little bit insufficient and highlights the need for advanced detection methods to capture this full spectrum of contaminants. And that leads us to the next part of this webinar, exploring how analytical strategies are evolving to meet these emerging challenges. And yeah, basically we could see that climate change is driving new mycotoxin risks, novel toxins, shifting hotspots, and complex co-occurrences. And as I said, no single method can really cover all these risks. So what we need is some kind of a toolbox approach, combining on the one side rapid field tools like biosensors and also lateral flow devices for a quick and on-site detection. But we also need new approaches like metabolomics and high-resolution mass spectrometry in order to detect complex, masked, and emerging toxins, and also try to predict contamination. And in the end, we also need some kind of the gold standard when it comes to the quantification of mycotoxins, LC-MS, for the accurate quantitative analysis, for the confirmation, and also the regulatory compliance. And together, these methods provide a flexible and layered monitoring system. And in our last webinar, I already introduced most of these methods, but what's new today are biosensors. So biosensors using a biological element, such as an antibody, an enzyme, or nucleic acid, to recognize the mycotoxin. When the binding occurs, it triggers a biochemical change that the transducer converts into a measurable signal. So this signal is then amplified and processed to give a quantitative or at least a semi-quantitative feedback. So we can say that biosensors are fast, field deployable, and also highly specific. They can detect multiple toxins at once and also help to guide targeted laboratory confirmation. The question is now only, how do biosensors compare to other rapid screening tools, such as the widely used lateral flow devices? So from a technical perspective, biosensors offer a very high sensitivity, also specificity, and as I said, they can also detect multiple mycotoxins at once, so they have the multiplexing opportunity. However, they are not yet widely commercialized. Most remain research tools that require specialized equipment and training, and they tend to be more costly. So on the other side, the lateral flow devices are very simple, very robust, and also cheap. They provide a very rapid, easy to interpret result for routine field monitoring, helping farmers and also inspectors to make fast and evidence-based decisions. So in summary, while the biosensors are definitely powerful tools for this rapid and precise research, the LFDs remain the standard for field monitoring and immediate decision making. But now I would like to shift gears a little bit and go beyond reactive detection. We also need to understand what actually drives mycotoxin formation. And by combining climate data, crop metabolomics, and artificial intelligence models, we can today predict where and when risks will emerge. So how does that work? Metabolomics, for example, identifies early biomarkers of fungal infection, often before toxins even appear. The artificial intelligence then links these signals with environmental factors such as temperature, humidity, rainfall, and also cropping practices to forecast risk patterns across different regions. And by integrating field, molecular, and environmental data, this allows a very proactive monitoring, enabling early interventions and more resilient crop management. And building on the predictive power of these tools, real-world studies show how this actually works in the field. So in one field study, a deep neural network applied to maize in the region of northern Italy predicted aflatoxin B1 and fumonisin B1 contamination with an accuracy of around 75%. In another study, found that specific metabolites such as vesiculorin B and sterigmatocysteine strongly correlate with aflatoxigenic strains. And by combining these biomarkers with AI models, it improved the aflatoxin prediction up to 87% accuracy, which is already quite impressive. The results show that actually metabolomics provides us actionable molecular insights, and AI turns it into some practical data-driven prediction for a proactive field monitoring. And another very powerful layer in this topic is added by the so-called high-resolution mass spectrometry. I have explained the technical concept of the high-res within the last webinar. So if you remember, high-res measures ions with a very high accuracy, allowing the detection of basically unlimited compounds, including unknown and also modified toxins. And this actually helps to discover both expected and unexpected mycotoxin derivatives, which is extremely important as a fungi can produce novel metabolites under these changing environmental and climate conditions. And for that, I have here one example to explain it a little bit better. An example of a masked mycotoxin formed in plants, namely the malonyl glucoside conjugate of cereal enol. So briefly, how does this look? The high-resolution workflow starts with a full scan to record all the ions. It's then extracted, or the relevant peaks are extracted for the analysis. The parent ions molecular formula is then determined by using the high-res mass accuracy, and the isotopic patterns are then compared to some theoretical distributions. And with the high-res fragmentation, it reveals the structural detail, confirming key features like the malonyl glucoside conjugation. So in that case, high resolution actually complements the metabolomics and also these artificial intelligence tools by enabling the characterization, I would say more importantly, also the confirmation of novel mycotoxin derivatives. And with that, it also helps to reveal how plants respond actually to stress. So in wheat and maize, for example, defense compounds like flavonoids and also certain amino acids change under heat and drought, often even before visible symptoms actually appear. So high-res studies have shown that these toxin levels clearly depend on the interaction between the plant variety and also the environment. In other words, fungal biomass alone is actually a poor predictor of risk. So by monitoring plant metabolism, we have some sort of an early warning system that crops are entering a high risk phase for fusarium infection and also the toxin buildup. And last but not least, high resolution mass spectrometry also offers another, I would say, unique advantage, namely the retrospective analysis. So when the data are collected in the so-called full scan mode, it allows us to revisit past data sets to find new or previously undetected mycotoxins. With that information, we can link, for example, heat waves or droughts to shifts in hidden toxins and plant defense metabolites, revealing the environmental conditions that can trigger high-risk metabolic states. So by combining this historical data with metabolic fingerprints and environmental records, high resolution definitely supports this predictive and climate adaptive agriculture, which allows the anticipation of high-risk scenarios and also informed decision-making, even for toxins which were initially not even monitored. Okay, so we have seen now several tools for the mycotoxin monitoring, like the biosensors for very sensitive multiplex detection, metabolomics and AI to predict emerging risks, and also high-res for the discovery of novel and or toxins, and also to provide this kind of retrospective analysis. Now we come to what I always call the gold standard for quantitative and regulatory grade monitoring, to the LC-MS-MS. So the advantage of such a triple quadruple mass spectrometer includes the possibility of tracking multiple mycotoxins, even in very complex matrices with a very high sensitivity and also robustness. So validated methods ensure highest accuracy and also compliance, while integrating data over time help us to link climate variabilities to toxin occurrence and also to co-contamination hotspots. In short, the LC-MS-MS turns insights from rapid tests, metabolomics, and high-res into actionable high-throughput data, forming the backbone of climate-smart mycotoxin control. And one of the most impressive and I would say comprehensive methods for mycotoxin testing in the field was actually developed by my former colleague and operational supervisor of my PhD thesis, Michael Suliuk at BOKU Vienna. This impressive method covers more than 700 mycotoxins and other secondary metabolites, making it the most extensive multitoxin approach currently in operation on this planet, I would say. And it's based on a very generic extraction workflow with a 5 gram of sample weigh-in, which is then extracted with an acetonitrile water mixture, which is slightly acidified. And then this extract is simply diluted in a ratio 1:1 with a complementary dilution solvent prior LC-MS-MS analysis. And this method basically applies a low injection volume of 5 microliter and a relatively high flow rate of around 1 milliliter per minute, which in this case provides a robust compromise between the chromatographic conditions and also the performance and the sample throughput. And methods like this are, I would say, the true workhorses out there in the field of mycotoxin testing, translating the lab precision into some real practical high throughput monitoring. And a key step for this kind of robust multitoxin LC-MS-MS methods is extracting these analytes from also complex matrices. So a landmark study in this case was conducted by Hans Mohl. He's a senior researcher from the Wageningen University in Netherlands in the year 2008, conducted this study where he laid the foundation for widely used dilute and shoot approaches, which I just mentioned. So Hans Mohl systematically tested multiple extraction protocols across pesticides, wet drugs, plant toxins, and also mycotoxins, and he actually figured out that partitioning methods like catchers often lost polar but also acidic or basic compounds, whereas acidified solvents like acetonitrile, methanol, or acetone with a low proportion of formic acid achieved the best results in terms of recovery. This actually showed that abandoning partitioning steps and lowering the pH reduces this matrix interaction significantly, and also enabling a simple and robust dilute and shoot protocol, which can be seen as the backbone of these large-scale LC-MS methods nowadays. Catchers, on the other side, which stands for quick, easy, cheap, effective, rugged, and safe, was actually developed by Anastasiadis many years ago, is another extraction method which combines this previously mentioned partitioning step, where you have at the end your purified extract, your adsorbents, and your catcher salts at the end of this layer. It is widely used in the pesticide area, especially as a cleanup tool, so to speak, to remove also matrix interferences, but it also adds some more complexity in your workflow when you compare it with the even more straightforward and simple dilute and shoot approach. So when we now move from the extraction to the detection, we have one key challenge when we talk about LC-MS, and this key challenge are matrix effects. So these effects are basically triggered by co-extracted compounds like salts, sugars, or other metabolites, which actually interfere with the ionization within the ionization process by either suppressing the signal, or in some cases it can also enhances the signal. Triple quad methods by following an electrospray ionization. In this case, analytes and matrix components enter the spray together, coming from the LC system. And when the matrix component dominate, they compete for the charge. That means they reduce the analyte ionization, and that produces at the end smaller signals. Some compounds can also enhance the signals by stabilizing charged droplets or also improving ion release. It is important to mention that these effects are very analyte and matrix specific. Generally, matrix effects can be classified as absolute and relative matrix effects, whereby the absolute effects compare a standard in a pure solvent versus, for example, a spiked sample. Relative effects, or matrix effects, are the so-called lot-to-lot variation, which occur between different batches of the same matrix. And because suppression and or the enhancement can distort the results, it is essential to address and actually to minimize matrix effects within the LC-MS analysis workflow. And this was actually a focus of one of my own earlier papers, where we systematically compared different cleanup and dilution strategies, and the results are summarized here in this graphic. In this case, each colored dot represents an analyte, so the colors are marking different approaches, like the catchers, in combination with different cleanup salts like PSA, which stands for primary secondary amines, but also C18 salts, as well as in combination with freezing step. This you conduct in order to precipitate lipid components, and also finally the different dilution levels ranging from 1:1 to 1:100. And what you see here in this picture is a quite clear trend. The stronger the dilution, hereby represented by these greenish yellowish dots, the smaller the matrix effect. So both for absolute and relative. So the y-axis you have the relative, and on the x-axis the absolute matrix effects summarized. And the reason is quite simple. So by diluting the extract, we also dilute these unwanted co-extracted matrix components. So reducing their concentration also reduces the competition, if you want. So the analyte signal is no longer suppressed or enhanced. In other words, dilution lowers both the analyte and also the interfering species equally. But since matrix effects are concentration dependent, the suppression becomes less pronounced. Catchers, on the other hand, aims to remove matrix interferences through these partitioning steps, what I already mentioned earlier, and the sorbents which are applied. It does help, but we also observed some losses for some certain analytes, especially for the more polar ones like nivalenol, but also monoylformine, but also the loss of fumonisins in combination with the PSA salts. So this makes it less suitable, if you want, for this kind of broad multi-methods. And the most practical compromise turned out to be this simple 1-to-1 dilution combined with a very relatively low injection volume of this 5 microliter and a quite high flow rate of 1 milliliter per minute. And this setup basically minimizes the matrix effects to an acceptable level while still retaining sufficient sensitivity for routine monitoring. So I think the key message here is that even small method adjustments can really make some big differences. And in the context of large-scale control, simplicity and robustness often outweighs chasing maximum sensitivity. And up to this point, we have looked at extraction and how to minimize matrix effects. But for reliable mycotoxin control, I would say this is only half of the story. The other cornerstone is the validation of these kind of big methods. So traditional validation schemes were actually developed for single analyte combinations methods in relatively, I would say, simple matrices. So when we move to multi-class, multi-toxin methods in feed analysis, the situation becomes more and more complicated. Feed, as such, is not a uniform matrix. So it is essentially a whole universe of different matrices, each with its own chemical fingerprint. Chicken feed is still somewhat predictable, I would say, with a quite high proportion of maize plus some smaller proportions of wheat and soybean. That's it. Pig feed, on the other side, is far more complex. So it may already include some diverse additives by byproducts and also sometimes even some challenging ingredients like blood meal. And cattle feed can vary really dramatically, depending also on the developmental stage of the animal. So ranging from pure concentrated mixture with high fractions of corn to really forage rich rations with high content of silages, for example. So the major problem is here when a laboratory receives a sample simply labeled as feed. In reality, this could basically mean everything. And a major part of my own PhD thesis was dedicated to this problem. So how can we actually validate multi-toxin methods in such an heterogeneous field, which standard guidelines do not reflect at the moment? And to address this, we have systematically studied both single feed ingredients such as maize, soy, barley, or silages, and so on, as well as a very wide range of complex compound feed. So for each type, we have evaluated matrix effects and extraction efficiencies across multiple lots in order to capture the variability that labs are facing also in reality. And this work actually laid the foundation for a new validation concept that better reflects the true diversity of feed. So what we did, we developed a validation framework tailored for feed. I would say actually for the entire feed chain. The idea was simple but powerful. So rather than validating against every possible feed variation, which is simply impossible, we created so-called model matrices that realistically simulate the three most important complex feed categories, including chicken, pig, and cattle. Together, these are covering more than 90% of the global feed production when we exclude the pet food sector. So what we did, we built these model matrices from the relevant raw materials in realistic proportions, and then included multiple lots per feed type, up to 7 different lots, to capture also this natural lot-to-lot variability that labs actually have to face in their daily business. And this made the validation process both scientifically robust and also quite resource efficient. And the results that these model matrices showed that these model matrices behaved actually similar to real-world compound feed, providing an excellent representative for validation experiments. So based on these results, we have then proposed a validation approach that prioritizes robustness and reproducibility across different feed types, rather than focusing solely on absolute recoveries. So while a wider range than classical single analyte guidelines is accepted in terms of the recovery, precision is maintained within strict limits. So the RSD should be lower 20%. And in other words, an absolute matrix effect can exceed the typical 20 to 30% suppression or enhancement in the LC-MS system. What matters is that these effects, together with the extraction efficiencies, remain consistent and reproducible across samples. And this actually ensures that even with larger matrix influences, the overall method performance is reliable and the result can be interpreted with a very high confidence. And this work was later then actually recognized by the American Chemical Society with the Best Research Article Award in the area of agrochemicals in the year 2021, underscoring again the impact in the field. And how this method also is working in the field also served here kind of the basis to collect some robust data in order to explore also how climate variability shape mycotoxin profiles. So by integrating these data with multivariate statistics, it reveals how environmental conditions influence fungal metabolism and also toxin combinations. In a study from also my former colleagues at BOKU, including more than 100 Austrian wheat samples collected between 2020 and 2021, 47 metabolites were detected, and 93 of which varied significantly with year, region, or both. So nearly 40 metabolites showed very strong correlations with rainfall and temperature, and the multivariate analysis, in this case conducted via PCA or ASCA, revealed really distinct patterns of multi-mycotoxin occurrence, which is linked to climatic variability and also shifts in dominant metabolites across years and also the sites. So this actually shows that LC-MS is more than just a quantitative tool. It also provides actionable insights for adaptive control, but also predictive risk assessment, and to help to better understand how climate change actually affects the fungal metabolite landscape. Okay, so after mastering now the big topic of multi-mycotoxin LC-MS in the lab, we also need these rapid on-site tools like the lateral flow devices. I've mentioned it already at the beginning of the analytical part. And under climate-driven toxin peaks, so to speak, these tools act as a critical first line of defense, which is can be directly applied in the field, enabling a faster and especially evidence-based decision. So lateral flow devices or tests are no longer simple yes or no decisions or devices. The modern versions provide already semi-quantitative and very robust results, even under challenging field conditions. So we can see that in a study from 2021 that shows, for example, in sub-Saharan Africa, samples that aflatoxin LFDs maintained over 86% accuracy, even under high heat and humidity, correlating closely with LC-MS results. Handheld smartphone-based fluorescence LFDs for ochratoxin A in wheat further demonstrated the potential for some semi-quantitative on-site analysis, achieving very satisfying recoveries and also reproducibility results. And moreover, LFDs with so-called nanoparticles boost the detection sensitivity by strengthening the signal from each bound analyte with, for example, gold or fluorescent nanoparticles, which are commonly conjugated to antibodies. So when the target mycotoxin binds, these nanoparticles accumulate at the test line, producing a stronger optical or fluorescent signal. And this high surface area and signal amplification provided by these nanoparticles mean that even very low concentrations of the toxin can produce a detectable response. And this allows earlier detection and also improves reliability, especially in challenging field conditions. So following these recent advances in lateral flow testing, commercial products such as the TotalTox and QuickScan from EnviroLogix now provide truly field-ready solutions for rapid mycotoxin assessment. So these tools actually deliver near instant on-site results for major mycotoxins, including aflatoxins, deoxynivalenol, fumonisins, and often within only an 8-minute workflow. The TotalTox, for example, enables simultaneous multi-analyte testing using single standardized extraction protocol, which also reduces the operator error and also shortens the training requirements while still maintaining robust and reproducible results. QuickScan, on the other side, further enhances the efficiency with automated reading, barcode tracking, and also data capture, supporting proactive decision making and actually also seamless auditing. So in regions facing environment or climate-driven mycotoxin peaks, these tools provide immediate field-level interventions, for example, at the point of harvest, and by delivering a rapid first alert, they allow farmers and also quality managers to mitigate contamination risks before they actually escalate. So while these lateral flow platforms already provide rapid on-site screening, recent events also highlight why such agility is increasingly critical. So climate change has been linked to a rise in aflatoxin risk, like we have seen in Serbia, where for the first time contamination was also detected now in soybeans, a crop historically considered at low risk. However, this shift actually illustrates how environmental change can actually alter the risk profiles of safe commodities, requiring continuous re-evaluation of monitoring strategies. So this case actually highlighted by the European Rapid Alert System for Food and Feed. It shows the need for a fast and reliable testing, and EnviroLogix responded by using its rapid validation methods to develop a quantitative aflatoxin test specifically for soybean. And by validating the assay with naturally contaminated samples rather than relying solely on spiked controls, high accuracy and also robustness were achieved, setting a new standard for field-adapted mycotoxin testing. Furthermore, this rapid field-level LFD testing is now also integrated into a broader mycotoxin management system at EnviroLogix. So this approach combines on-site LFD screening with ISO 17025 accredited LC-MS lab confirmation for the big 6 mycotoxins, with results delivered within only 4 days. So that's really fast. And this basically ensures that initial alerts from the field can be quickly verified in the lab, bridging speed and accuracy in this case. The certified quick check reference materials play also a crucial role in this ecosystem. So they basically standardize operator training, but also monitor equipment performance and provide audit traceability, which ensures that both field and lab results are reliable and reproducible. So furthermore, we have centralized data collection through the so-called Total Hub Cloud platform, which allows real-time visualization of trends adaptive testing schedules, and also streamlined compliance management. In short, EnviroLogix provides a fully integrated mycotoxin management ecosystem that combines rapid field screening, reliable lab confirmation, standardized reference materials, and a centralized data management. And together, these components create a seamless workflow that actually transforms isolated measurements into actionable insights, enabling early warnings, informed supply decisions, and adaptive risk management under variable environmental conditions. And with that, I'm already coming to an end of this today's webinar, where we could see that climate change is already reshaping mycotoxin risks, driving shifts in geographic prevalence, but also in the co-occurrence of the patterns and the fungal metabolite profiles. So these changes have a massive impact on food and feed safety, requiring proactive integrated strategies like those from EnviroLogix. Rapid field level tools such as the TotalTox or the QuickScan LFDs provide first-line screening at harvest or storage, advanced lab analytics with properly validated LC-MS methods, and also untargeted high-resolution MS help to quantify multiple toxins at the same time, and also to detect hidden metabolites and link contamination patterns to environmental and climate factors. Coupled with robust validation frameworks, these approaches ensure reliable results even in highly variable matrices like complex feed. Integration is critical. So platforms like TotalHub connect field data with lab confirmation and also centralized analytics, so enabling early warning, adaptive interventions, and also informed supply chain decisions. Looking ahead, future scenarios include more frequent severe climate-driven contamination events, and by combining environmental monitoring with predictive analytics, as well as digital traceability and high-resolution detection tools, we can create a resilient, data-driven system that actually safeguards agriculture and also protects public health and ensures the regulatory compliance. A true convergence of climate science, farming practices, and also analytical chemistry. And with that, I'm at the end of this webinar. Thank you very much for listening, and I will be now ready to answer some questions